Fact 1. Beneath our skin, mucous membranes, and fascias lies not just connective tissue padding, but a gigantic network of fluid-filled microcavities, the interstitium. Its scale has only truly come into view in recent years thanks to live tissue endomicroscopy. Classical histology sealed samples with fixatives, causing the cavities to collapse, making the entire system appear as a dense matrix. In the living body, however, the interstitium extends as a continuous sponge-like network beneath the digestive tract's epithelium, within the walls of the urinary tract, near the bronchi, between muscle fibers, and under the dermis. Collagen and elastin fibers form reinforcing bridges between which fluid circulates, collecting immune cells and inflammatory molecules and directing them to lymphatic vessels. It's not a single sac, but a shock-absorbing system. It takes mechanical impacts from walking, coughing, and peristalsis, distributes pressure, and softens microtraumas. For oncologists, the discovery has proven alarmingly useful. The interstitium serves as a rapid bypass for metastases, allowing tumor cells to slip far from the primary site. For clinicians, it's a new source of biomarkers. It's here that readings can be taken before changes enter the bloodstream. There's also a common explanation for familiar phenomena. Age-related wrinkle patterns and swelling involve not only the skin and vessels, but also the interstitium's tired drainage sponge, whose framework collapses. Calling the interstitium a new organ is debatable, and scientists acknowledge this. But at least we've gained a large-scale, previously hidden layer of anatomy, without which the picture of the human body was incomplete. Fact 2. In the cortex of the human brain, a special type of inhibitory neurons not found in laboratory mice was discovered. Rose-hip neurons, sometimes called rose-hip in Russian due to characteristic thickenings, berries, on the axon endings. These cells are compact yet intricately branched. Short dendrites collect local signals from the upper layers of the cortex, and the dense, swirling axonal crown envelops the distal branches of pyramidal neurons like lace on a cuff. Electrophysiologically, they produce short, precise inhibition pricks. They don't drown a whole column, but fix the necessary millimeter of the algorithm. According to transcriptomic passports, this is a separate class. Sets of activated genes and receptors differ noticeably from known subtypes of inhibitory cells, which allowed them to be isolated in the single-nucleus RNA sequencing array. Anatomically, they are found more in layers 2-3 of the associative cortex, where the brain combines contexts, compares, and refines. Hence the tempting hypotheses. Such neurons might be responsible for the ultra-fine tuning of human speech processing, abstractions, emotional tones, for what doesn't scale well from mouse to human. The practical conclusion is prosaic and important. Models of psychoneurological diseases created on rodents may miss critical mechanisms. If rosehip cells participate, for example, in the inhibitory control of cortical waves, a malfunction in their work could make an invisible contribution in mouse experiments to epilepsy, autism spectrum disorders, or depression. The new cell is not just a rare branch on the tree of neuroanatomy, but the key to why the human brain thinks this way. Third fact. In 2020, the fourth pair of salivary glands made headlines, hidden where you least expected, high in the nasopharynx on the torus tubarius near the opening of the eustachian tubes. It was seen not with a scalpel, but with light. Doctors were looking for prostate cancer metastases using PET-CT and a radiotracer that accumulates intensely in salivary tissue. In the images, two symmetrical tubular structures several centimeters long suddenly flashed. An anatomical study on cadaveric material confirmed. These are not accidental islands, but continuous, richly innervated glandular ducts, closer in structure to the large salivary glands than to the usual scattered small glands of the mucosa. The function is to lubricate and protect the posterior wall of the nasopharynx and the openings of the tubes, so that swallowing, breathing, and talking proceed without friction. For head and neck radiation therapy, this discovery proved practically important. If these areas fall under the beam, patients more often experience dryness and pain when swallowing, which is why radiologists began discussing new organ preservation contours to more carefully bypass the tubarial glands. However, debates continue. Some anatomists believe this is not a new pair, but a large conglomerate of small glands that was simply seen for the first time so clearly due to the specificity of PET markers. Consensus is forming slowly, but the essence is clear. The map of the human salivary system was incomplete, and the new detail is already influencing practice, from radiation planning to explaining why some patients have persistent dry throat after a cold while others recover without a trace. Fourth fact. It was recently discovered that pain of mechanical origin does not start solely from the naked nerve endings of the skin. Beneath the epidermis, a very fine network of special-type Schwann glial cells has been found. They envelop nociceptors and are themselves sensitive to deformation, essentially forming a separate organ of pain perception. In animal experiments, optogenetic activation of these cells triggered typical protective reactions as if the skin had been pricked by a needle. And when their activity was suppressed, the pain sensitivity threshold notably increased. A morphologically similar network has been identified in human skin, and clinicians increasingly view it as the missing link in hyperalgesia and allodynia, when even light touch burns like sandpaper. History is rewriting the textbook. Glia are not just the support staff of neurons, but an active sensor and amplifier. This explains why local inflammations and scars cause phantom pain. It's not just the nerve wiring that changes, but also the sensitivity of the glial network. The practical takeaway is simple and encouraging. Targets for pain relief are shifting to the periphery. If we learn to finely modulate the activity of these Schwann cells using drugs, light, ultrasound, or electricity, pain can be relieved without stunning the entire brain with sedatives and risking opioid dependence. For now, this is a research path, but the map has been unfolded. Beneath the skin of each of us operates our own signaling web, which is the first to decide what exactly counts as a dangerous touch. Fifth fact. Over the last decade, the brain's drainage system has been reevaluated. It turns out that it has a complete drainage system, the glymphatic system, which works in conjunction with the recently mapped lymphatic vessels of the dura mater. Cerebrospinal fluid not only bathes the brain's convolutions, but is pumped along arteries into perivascular tunnels, driven by vascular wall pulsations. It flushes metabolic waste products from the interstitial space, carrying them to the veins and further, through meningeal lymphatic collectors to deep neck nodes. The key detail is sleep. During the deep slow wave phase, neuroglia adjusts the tuning of water channels. Intercellular spaces widen, estimated by tens of %, and the flow dramatically accelerates. MRI scans in humans show waves of cerebrospinal fluid moving synchronously with cortical fluctuations. This leads to practical implications. The brain's nightly wash is not a metaphor, but physiology, and chronic sleep deprivation is linked to higher beta-amyloid accumulation and impaired clearance of inflammatory molecules. Posture is also important. In animal experiments, the lateral position improved outflow compared to lying on the back, a detail currently being cautiously tested in humans. Another aspect involves vascular pathologies and trauma. Microbleeds and arterial stiffness disrupt perivascular pumping, and dural edema can compress meningeal collectors. In essence, we are faced not just with an interesting scheme, but a new therapeutic target. From the rate of slow-wave sleep and respiratory synchronization to gentle stimulation of vascular pulsation, all are being considered as ways to help the brain better clear waste. Sixth fact. Bone is no longer the body's concrete. In the cortical layer of long bones, a network of transcortical vessels has recently been described. Thousands of microchannels that literally pierce the compact tissue from the bone marrow to the periosteum. On transparent corrosion casts and high-resolution images, it is visible how part of these tubules connect to the arterial inflow and part to the venous outflow. Cells and molecules continuously circulate through them, connecting the closed cavity of the bone marrow sinus to the superficial vascular network. This explains two old clinical puzzles. Firstly, why intraosseous access works lightning fast when veins cannot be found. The drug exits through these shunts directly into the main arteries. Secondly, why inflammation in a joint so quickly raises bone fever. Leukocytes and cytokines get a direct route from the bone marrow to the focus and back. New traces have also emerged for rheumatologists and oncologists. The remodeling of these channels accompanies arthritis and fracture healing, and in oncology, it is discussed whether they serve as a bypass for metastatic cells. The wording is still cautious, but data is accumulating. For skeletal physiology, this is a revolution. Compact bone is not an inert wall, but a living tissue permeated by a metro network, where blood flow, immune traffic, and mechanical loads literally meet at one point. And for practice, new targets, from drugs affecting the endothelium of these microvessels to imaging methods capable of showing in advance where bone is healing and where it is stagnating. Seventh fact: the textbook on the human genome is being rewritten. Instead of a single standard, we have obtained a pangenome, a graphical map compiled from dozens of complete genomes of people of diverse origins. Furthermore, it was assembled not from short snippets, but through long-read sequencing that traverses repeats and complex DNA loops. It turned out that the previous reference genome, while convenient, was one-sided. It poorly captured large insertions and rearrangements, characteristic of some populations and rare in others. The pangenome has added about hundreds of millions of missing base pairs and thousands of structural variants to the picture. It has completed the labyrinths in the HLA immune complex, clarified the copy number and breaks in the memory-laden SMN1/SMN2 gene pair, and highlighted the accordion of repeats in LPA, linked to the risk of atherosclerosis. The key is the graph format. Now, normal is not a single thread, but a network of paths along which a clinician can match the genome of a specific patient without an imposed Eurocentric trajectory. In practice, this is already changing screening. Rare variants in inhabitants of Africa, Oceania, or indigenous peoples of the Americas are no longer lost in blind spots. And personal risk assessments, polygenic risk scores, are starting to work not only for those on whom the model was trained. Engineering has also benefited. Alignment algorithms learn to walk along the graph, and diagnosing repeat expansions is no longer guesswork. The reverse is also important. The pangenome does not close the topic. It is a living project where the library is continually augmented with new assemblies and contentious regions are refined. But the lesson has already been learned. The human genome is not a single book, but a multilingual archive. And only by reading different volumes do we begin to understand why the same diagnosis behaves differently in different people. Fact number 8. Within your blood can live for years silent clones, hematopoietic stem cells with an identical, fortuitously advantageous somatic mutation. This phenomenon has been termed clonal hematopoiesis of indeterminate potential, or CHIP. The scenario is simple and insidious. A bone marrow stem cell accidentally suffers a glitch in its epigenetic regulator, most often TET2, DNMT3A, ASX1, sometimes JAK2, causing its descendants to divide slightly more confidently than their neighbors and gradually occupy a noticeable share of your blood formation. The person feels healthy, but the composition of their monocytes and macrophages is already skewed. Such cells more actively switch on inflammation programs, broadly distribute IL-1 beta and IL-6 signals, and more readily feed arterial plaques. According to observations, in 10 to 20% of people over 60, such clones can be detected in the blood, and their presence is associated with a higher risk of heart attack and stroke, with accelerated vascular calcification, as well as with unclear outcomes after heart surgery and the course of heart failure. However, CHIP is not cancer. In the majority of carriers, the clone remains silent, and the risk of transforming into leukemia is estimated to be low, at fractions of a percent per year. The practical turn has already begun. Cardiologists and oncohematologists are attempting to include deep blood sequencing in risk assessment, and the therapeutic focus is shifting towards containing the inflammatory echo of clones, from targeting the IL-1 beta pathway to the careful use of old anti-inflammatory regimens. Another facet is prevention. Tobacco smoke and chronic inflammation seem to fuel the selection of such clones, while risk factor control and good sleep, conversely, deprive them of their advantages. In other words, aging harbors a genetic microdrama, and we are just learning to read it. Ninth fact: scientists have literally seen traces of civilization in us. Microplastic and even nanoplastic are now reliably found in human tissues. First in the placenta and cord blood, then in samples of lung tissue, lymph nodes, and later in the circulating blood of adults and in atherosclerotic plaques of the carotid arteries. These are not guesses or dust on the filter. Particles are captured by their chemical polymer fingerprint, pyrolysis gas chromatography mass spectroscopy, and by their optical spectrum, micro-Raman IR microscopy. Their size, tens of microns and below, and shape, fragments, fibers. How do they get there? Two main routes: air and food. Aerosolized fibers settle on mucous membranes and penetrate deep into the alveoli. Fragments come from water, seafood, salt, and migrate through the intestinal barrier, sometimes under the guise of a protein corona, to which immune patrols adhere less. Inside the body, plastic behaves like an irritating foreign body. Macrophages engulf it but do not digest it. Low-grade inflammation is triggered around it, and oxidative stress pathways are activated. Hence the cautious but worrying associations. The more particles, the more frequently unstable plaques and microthrombotic inflammation are detected. In pregnant women, findings in the placenta are discussed as a potential factor for metabolic disorders. It is important to understand the limits. A direct causal relationship for most effects is still being proven, and methods are being standardized to exclude laboratory contamination. But the fact itself has already changed the perspective. Humans have proven permeable to synthetic dust, and 21st century toxicology now looks not only at molecules, but also at the tiny bodies of materials, their size, charge, shape. Practical conclusions emerge immediately, from filters in ventilation and water treatment systems to the review of food contact packaging and regulations for dust-generating industries. 10th fact: aging has exceptions, and neurobiologists have only recently begun to dissect them. Superagers, people over 80 with memory comparable to much younger individuals, are not just lucky gene carriers, but possess a special architecture and purity of their cortical circuits. In comparative studies, they have thicker cortex in the insula and anterior cingulate, stronger connections within the attention network, and more functional frontotemporal loops, which keep distracting stimuli in check. Most striking is the microscopy. In the entorhinal cortex, critical for fixing episodic memories, superagers have large, healthy pyramidal neurons in layer 2 with less tau pathology burden and better preserved dendritic trees, the spines where synapses set. This is not immortality. They also undergo age-related restructuring, but their starting reserves are higher, and protein junk accumulates more slowly. The behavioral picture matches the anatomy. Superagers switch attention faster, lose the thread of conversation less often, and recall details more accurately. What's the source of the advantage? The mosaic is composed of genetic factors, vascular health, long cognitive load, and, as imaging studies show, a stable sleep architecture with generous slow waves, fueling the brain's nightly cleaning cycle. The practical meaning is not that anyone can do it, but that the human brain is potentially capable of aging asymmetrically, and this opens a window for interventions: attention training, careful cardiology, fighting inflammation, managing sleep deprivation. Superagers have become living treasure maps. Following their pathways, neuroscience now seeks concrete, measurable ways to strengthen memory bridges rather than just regretting the destroyed ones.